agenda it is, and I have 4 topics. And the first, drug discovery is a long way journey. As you know, it takes 9 to 17 years from the target identification through market launch. The further success rate of the drug discovery is very low. Only one compound among 20 to 30,000 compounds can be launched. Actually, only 4 small molecules and 6 non-small molecules was approved in Japan in 2019. Even in US, in the same year, about 40 small molecules and 10 non-small molecule new drugs got approved. So improvement of drug discovery process is critical to be solved. And there are 3 key success factors for drug discovery: target and molecule and translation. Selection of right target and selection of right modality and molecule, and evaluation of target and molecule with right process. So first, I'd like to talk about target. So a drug target is a molecule in the body is intrinsically associated with a particular disease process and could be addressed by a drug target and produce a desired therapeutic effect. Typically, a target is a protein. This figure shows target class of first-in-class drugs according to the molecular type and target family. As you see, there are various target class like GPCR, kinase, protease, ion channel, and other enzyme cytokines, etc. Then let's see the actual drug. This is the list of the best-selling drug in 2020. The Humira and the top seller earns $20 billion, and its target is a cytokine TNF alpha. So others, the PD-1 is a membrane protein, membrane receptor, and the protease factor 10, and the enzyme ubiquitin ligase, and the kinase VEGF alpha receptor and BTK, and so on. So actually, after drugs, best-selling drugs are also use various targets. So how many proteins can be a target? And druggable target for small molecule and that modify disease are 600 to 1500, literature says. So among them, launched the drug already used 300 to 400 targets. So I think still more than 500 targets remain undrugged. But among them, challenging drugs are included for small molecule. So then small drug discovery technologies for small molecules are being explored. In addition, in addition to small molecules, new modalities are emerging, like oligonucleotides and gene therapy and cell therapy. So I don't explain the details, but more than 20 products were already launched from these new modalities, and increasing the number of the new modalities. Yep. As I said, success rate of drug discovery is low, but this figure shows R&D productivity models. In terms of target numbers for drug discovery, this figure shows about 25 targets or 25 assets is needed to launch one product. Then even at the beginning of the clinical stage, so 9 assets is needed. So success rate is very low. It's kind of awful. So selection of right target at the beginning of the drug discovery is critically important. As you know, translational approach is taken. The conventional translational approach is to start with target of interest, and then go to the compound generation, candidate selection, and go to the development stage. But nowadays, to improve the success ratio of drug discovery, reverse translational approach is getting more important. But the reverse approach is patient first. So at the beginning, understanding of disease and patient is important, and start with disease and patient understanding, and go select at the right target for that disease, and start candidate generation and development and stage according to the translational approach, conventional translational approach. So this is the flow of target discovery, and a very simple flow. The target patient segment is at the beginning. Target patient segment is selected, but based on the business strategy and unmet medical needs, and the product image will be generated. This product image is a target, is the target product profile of the TPP. It's very important. The target product profile, TPP, outlines the desired profile of characteristic of the target product. And the TPP state various aspects, including intended use, target populations, and other desired attributes of the product. It's not only safety, but also efficacy-related characteristics. So, like this. The right table shows a actual TPP example for ischemic stroke revascularization. In this TPP example, the minimum, it may be small, letter is small, I'm sorry, minimum acceptable result and ideal result. Both results are set for the profiles. For example, patient population and delivery mode, dosage form, regimen, and efficacy and safety, side effect, and so on. The goal, TPP is the goal of the product. When the target molecule is selected, so target assessment will be conducted. The target assessment is to check whether that target molecule could potentially produce a target product that match the TPP. So the check is done, will be done from the viewpoint of causality, safety, feasibility, and strategy. Before starting actual drug discovery, I mean the screening, HTS. Let me talk about the details about the target. So this table shows the summary of desirable target drug target profile. The first one is disease modified. So target should be disease modified. I think this is the most important for therapeutics. And second one is less important under physiological conditions. That is for safety. from the safety viewpoint, and the druggability. If a 3D structure for the target protein is available, it will be very useful for actual drug discovery or drug design. And assayability. So even if the target is very good, the assay is not set up, assay is not feasible, we can't produce the drug. And not uniformly, target expression is not uniformly distributed, and the prediction of the potential side effect. This is also from the safety viewpoint. And a target-specific biomarker, if the biomarker is available, it enable efficient drug discovery from the efficacy viewpoint or from the safety viewpoint as well. The last one is IP situation. So favorable IP, intellectual property situation. It's not scientific, but very critical for business. So we have to consider about these points to select the drug target. I think I should be not about causality for target assessment, because a molecule that is correlated with disease, yeah, even if the molecule is correlated with the disease, it is not always a right drug target. For example, the figure shows HIF case, hypoxia inducing factor. So high levels of HIF is correlated with cancer survival. So high HIF patient shows poor prognosis. This is a clinical information, but there are several hypotheses or interpretation can be made. The first one is HIF directly induce aggressive growth. If this is the case, so HIF can be a good target. But other hypotheses can be made. So aggressive growth induce HIF. It's not cause, it's a result. And other hypothesis is hypoxia can induce aggressive growth, but HIF does not. So we have to consider these hypotheses and check the have to validate this hypothesis from the information, clinical information. As for target validation, usually we use animal models in many cases, but it should be careful to select animal model as well. So this is the example for IBD, inflammatory bowel disease models. There are 3 models summarized, DSS, and 2 T-cell transfer models. There are characteristics different among the 3 models. For example, T-cell transfer, activated T-cell transfer model is more inflammatory and chronic. And the anti-TNF is very effective. On the other hand, DSS model is less inflammatory and acute. So anti-TNF is not so effective, but surfacing is effective. So that means animal model is not human diseases. So efficacy of the compound should be evaluated in a model in which that MOA, mode of action of the compound, contribute to the pathophysiology. Let me introduce one example for actual target discovery. It's for IBD, and as you know, TNF, anti-TNF antibody or anti-TNF agent is very effective in clinic. But even in responder, TNF responder, that the patient is not completely healthy or normal at the level of molecule. So to cure IBD, a new target was investigated. So I don't talk about the details, but the approach is by using bioinformatic analysis and clinical information. So gene expression, gene signature. That means comprehensive gene expression profile of responder, TNF responder and healthy subject are compared. And then pathway or the molecule target investigated that reverse the gene signature from responder to healthy. And finally, RAF-MEK pathway was highlighted like this. So comparing gene signature and one pathway was identified. Then next step, the validation was done using IBD model. As you see, MEK inhibitor was used in this study. As you see, MEK inhibitor improved clinical score in the IBD model, and also clearly improved pathophysiology like this. So the hypothesis of MEK inhibitor was validated. So in the preclinical stages. So further investigation for drug discovery is expected for this target. Let the summary of the target discovery to be as a point to be considered. So disease understanding is very important, and the TPP will guide the disease understanding and the selection of the target. And the molecular level of understanding of MOA, it is needed to check relevance to the human disease from preclinical research. And the third one is access to model to technology. So that is needed for actual generation of drug candidates. Then I would like to move to beta molecule section. Even though we can select ideal target, but we can, if we cannot generate a right molecule, we can't deliver a drug to the patient. This table summarizes the primary cause of the failure in drug development, the actual failure. So there are three major causes for the failure: clinical and non-clinical safety issue, especially in the non-clinical stage and phase 1 stage. In addition to safety, the efficacy and pharmacokinetics and bioavailabilities are major cause. For the efficacy and the major cause of the efficacy, major cause of failure is efficacy in the phase 3. So we have to check three profiles for a good drug. We call it drug likeness. So efficacy, safety, DMPK. The bioactive compound is not the drug. So efficacy, safety, DMPK profiles are required for a drug. A deviation from drug likeness causes the risk of clinical development failure and early withdrawal even after the launch. Inferiority of in drug likeness will face the competition with drugs that are similar to less potent in efficacy. So it's less competitive and causes less competitive. So drug likeness is to be evaluated from very beginning of the drug discovery. In the course of drug discovery, lead optimization will be is done with multiple parameters. And the structure activity relationship, SAR, and the structure property relationship, SPR, is conducted, is analyzed in parallel with lead optimization stages using these various parameters, in vitro and in vivo parameters, including biochemical, cellular potency, efficacy, PK/PD, and toxicity, etc. I will touch on a little bit more detail of the assay. So as for the ADMET assay for DMPK tox evaluation, the physical chemical properties, absorption, distribution, metabolism, excretion, and toxicity profile are evaluated using these assays or parameters. These assays may be a little bit different company by company because of the company strategy. So, but it's generally these parameters are used for ADMET. For safety evaluation, the same various safety evaluation is done will be performed based on the stage from hit to lead or to lead optimization. So I would say any candidate have some safety issues, but there are several critical questions. So what is the safety margin? How large is safety margin? So acceptable safety margin is depend on the indication or profile of those limiting adverse event or complete fluctuation and so on. And is the toxicity reversible? Reversible. So toxicity that is irreversible is typically unacceptable. So killer toxicity. And is there a biomarker? Toxicity cannot be monitored, may develop into irreversible toxicity, so killer toxicity. And what is the mechanism, and what is the relevance of the finding to humans? So we have to check the toxicity profile observed in the animal is species specific, for example, rat specific or mouse specific or not. So considering these questions, the evaluation, risk-benefit evaluation will be done. The efficacy evaluation, as for efficacy, to find pharmacodynamic activities can be detected with exposure of the drug at the target site is evaluated. So PK/PD analysis. And the other important point is target engagement. So binding to the actual target is really important because if it doesn't show the target binding, it may cause the off-target effect and the safety concerns. The third one is target modulation. So we have to know actual proof mechanism of action. It is really important to understand the actual efficacy is human relevant or not. And again, these are three key success factors for drug discovery: and target, the molecule, and the beta translation. So if we evaluate these three key factors, and if we use these three key factors, innovative drugs will be efficiently generated. At the end, let me introduce our company very briefly. We Axcelead Drug Discovery Partners is a solution provider in Japan. So it established in 2017 with the Takeda spin-off company, and currently fully independent. Employee number is about 270, and located in Shonan area near 1 hour train ride from Tokyo. And up to today, more than 150 organization is our customer, and getting increased. And as I said, we are a spin-off company from Takeda, and we are inheriting Takeda's unique property, unique and proprietary drug discovery platform, people, infrastructure, and historical data, and have capability for small molecules and peptide drug discovery, from target discovery through preclinical development, even breeding process to the clinical development. And also, we are pursuing partnership for new technology and strong relationship with Japanese government as well. So even if we have full capability, we have to continuously pursue the new technology to provide a better service for our clients. Thank you so much.